All right, we're going to walk through the answers to the eukaryotic gene regulation reading assignment that you did out of your textbooks, right? Section 15.2. Okay, so just going to walk you through so you can hopefully figure out how you did, get some more explanation to that. So question 1 about histones and nucleosomes. So basically what happens here is DNA gets wound around these little proteins called histones, okay? And then the proteins, the histones, form these clusters. And when you have a cluster of its usually 8 histones, it's called a nucleosome. And then these nucleosomes get all like packed together, and these things all coil up to make a chromosome. So it's just kind of more DNA organization, really, right? We have DNA get wound up so that it doesn't get tangled, or that it can get organized, so that when things are moving around during mitosis, that we don't have some DNA going this way, some going that way, it all stays together. And one of the ways that DNA does this is to wrap itself around histones, and then those histones kind of cluster together and coil even more to form a nucleosome, which then condenses into a full-on chromosome, okay? So we've learned also about, like, with operons, right, how things get turned off and turned on, okay? But with eukaryotes, they don't have operons, okay? So we're now going to learn about some of the ways that our genes can be turned off and turned on in a eukaryotic organism. One way is through acetylation, okay, histone acetylation. So these acetyl groups, okay, which is a chemical group, these little acetyls attach themselves to the histones. And these acyl groups, if you look down here, like, they stick out all kinds of ways. And so the histones can't pack as tightly when the acetyl groups are attached to it. So when the histones are acetylated, this is going to spread out our histones, which means look at all this DNA right here that's available to be transcribed. Oh, mRNA polymerase could get in there and do its work. So when we have lots of acetylation, we have our histones spread out really far, which means that this is going to turn on this, like, part of the DNA, and so it can get transcribed and then translated into a protein. Okay, so histones spread things out and turn things on, whereas methyl groups do the opposite. Okay, methyl groups attach directly to the DNA, though, not to the histones. But the methyl group, between the interactions of the methyl groups, is going to shrink things together. And so we'll see here the methyl groups are in red. So, and here is where things are acetylated, right? So if you take out these acetyl groups and add the methyl groups, we're going to really get things tightening up. And so this is going to make the DNA just, like, literally physically less accessible to RNA polymerase. It's not going to be able to get in there because it's just not going to physically fit. And so more methyl groups means less transcription and translation. So this would, like, turn things more off, whereas this one kind of turned things on. We're going to have lots of action with RNA polymerase and transcription and then translation. And here we're going to have little to no because it's just too tightly packed together. Okay, another good diagram just to show you. Question 4 then talked about epigenetics. Now you're going to, or maybe you've already done, a whole assignment about epigenetics, okay? So more on this. We're going to keep it vague for right now, but basically it's that you can have different individuals showing different gene expression, but keeping the gene, like, the DNA code stays the same. But basically, like, why are certain genes on in some people and certain genes off in other people? We have the same genes, but why are some people's on and some people's off? It has to do with epigenetics, that literally what happened to oftentimes our mothers or grandmothers and what happened there, like when we were growing in the belly, will influence our gene expression once we're out of the belly. And it's pretty crazy. And you're going to see some firsthand examples of this on your epigenetics assignments. Okay, so more on this to come. Number 6. Nope, number 5. So a eukaryotic gene map. So as opposed to having an operon with, you know, a promoter and an operator and a terminator, as you can see, some similarities, some differences. We still have a promoter. This is where RNA, you know, but it's not going to be quite the same, but RNA polymerase is going to attach here. We have these things, exons and introns. We have a poly A signal sequence and a termination region, okay? We also have these proximal and distal control elements. And what they are is they are segments of non-coding DNA that have a very specific sequence that this is going to serve as the binding site for our transcription initiation factors. Okay. And so if you think back to when we learned about protein synthesis, transcription, and translation, one of the things that make prokaryotes and eukaryotes different is that eukaryotes have a transcription initiation complex. Okay. And so here's the like zoomed in. This is what 7, 8, and 9 deal with. But basically, we've got this whole cluster of things that come together and like fold the DNA over and position it perfectly for RNA polymerase to get in. Okay. And so these parts right here are where these blobs are going to like join up. Okay. So that's why they're control elements, because in order for anything to happen, like things have to fold and line up perfectly. Okay. So those are our control elements there. We're going to have exons. These are parts, the way I remember between exon and intron, exons get expressed. So exons, these are like coding portions of DNA. Introns are non-coding portions of DNA. Exons coding, introns non-coding. Poly-A sequence, this is a whole bunch of A's all in a row that tells you, it's almost time. It's almost time to stop transcription now. And the termination region is where the RNA polymerase actually breaks free. Okay, so the RNA polymerase does bind at the promoter, but it's got to be, everything's got to be folded and bended over perfectly first. Okay, so here's a look at that. And so basically what happens is these different proteins match up with our sites, either our proximal or our distal, and all that means is the proximal are close to the promoter, the distal are further away. Okay, but we're going to have these helper proteins that bend it to get the perfect little cushion, the perfect little little pouch that RNA polymerase fits through, and our RNA polymerase is going to bind at the promoter and is going to zip along. Okay. One thing to know, another thing to know about this is that in this promoter is what we call the Tata box. Maybe you can tell why it's called the Tata box, but the Tata box is a whole, it's T-A-T-A-A-A. It's this whole sequence of T's and A's. And this is the part of the promoter that RNA polymerase latches onto. Okay. And so then once all of this is in place, we've had our activator proteins, the whole transcription initiation complex bends and positions everything. RNA polymerase finds that Tata box of the promoter. Whew, now we're ready. And it's going to unzip the DNA and it's going to start building our piece of mRNA. Okay. And then once the mRNA is made, then that gets taken to the cytoplasm and that can be translated into amino acids and making a protein. Right? Great. All right, next one here, control elements. So basically, if you think about it, all of our cells have all the same DNA, right? Like my nerve cells have the same DNA as my stomach cells, as my liver cells, as my eye cells. But you can imagine that there might be certain genes that like my eye cell is never going to need, but my stomach cell is going to need all the time. Okay. And the way that this happens is that in order for this gene to be transcribed, I've got to have the right things binding here right at this gene. Well, if a certain cell doesn't have those things, it's never going to bind. Okay? So if we're talking about something that a liver cell makes and needs, it's going to have the perfect, look, the yellow and orange, it's going to have the perfect control proteins in there. Okay, and they're going to help fold this over and transcribe this gene that's necessary in livers. Okay, but look at my brain. Does my brain have those like orange and yellow things? Does it have these two? No, it's got those ones. So my brain isn't really going to ever transcribe this, or really not at all, because it doesn't have the right things. It's never going to get bent over into this position, and so RNA polymerase isn't going to come in. So that's why you can have a gene in your eye or in your heart, but your cell will never make that thing because it doesn't have the proteins that will bind to these activation sites that will then get your gene in the right position to be transcribed. Okay, so these different regulatory factors will be present in different amounts in different cells. Okay. There's also a whole assignment on cell differentiation, so more on this to come. But basically, if a cell doesn't have these things, it can't turn this gene on. If it does, then it can. Okay. Number 11 here, we're looking at 3 genes that are expressed in muscle tissues. What would you expect to find? You would expect, because they're all muscles, you would expect all of these enhancers, you would expect those sequences to really be similar, because all muscle cells would probably need to be transcribing and translating that gene. Okay, so those sequences would be very, very similar. That's all we're getting to here. Because although there are 3 different muscles, they would probably need to be transcribing similar genes. Number 12, this is a really big, important thing to understand. This is a big concept that often gets asked about on the AP exam. And this is called alternative splicing. So like we saw on our little gene map here, right? We have these exons and introns. Exons get expressed. They actually code for a protein. Introns, not so much. So one of the things that you might remember also back from our transcription translation lecture was that in eukaryotes, the mRNA undergoes some processing. And I said the introns get taken out. Okay, so all those introns go away that leave exons. That's true. And also, sometimes not all the exons get expressed. So I could have this one piece of DNA. I'm going to take out all the introns because they're garbage. They're nothing. But over here, I leave in exons 1, 2, 4, and 5. Okay, I forget exon number 3. Then I'm going to take that piece of mRNA. It's going to get translated. It's going to make a specific protein. But then over here, I could take the same piece of DNA, get out the garbage introns, but this time I'm going to skip exon 4. I'll leave 1, 2, 3, and 5. That's going to make a different protein that does a slightly different thing. Okay. And so you can imagine, what if I left out 2? What if I left out 1? What if I left out 5, right? Like you can imagine all the different combos of these exons could all produce slightly different proteins that are going to have slightly different functions. And that's why we can make so many more proteins than we have genes, because of all the different combinations we have of these exons. Okay, and then lastly, a way that your cell can control how much protein is in the cytoplasm. If you remember back to our protein lecture, one of the things that happens to mRNA processing is we get the cap and the tail, okay? And so one of the things, we've got enzymes in the cytoplasm, and we're looking right down here. Oh, there goes the bell. We've got enzymes in the cytoplasm, and these enzymes, as soon as they find mRNA, they don't know if that piece of mRNA has been used or not, and your cell is really efficient, and it wants to break stuff down that's not being used. So it doesn't know if it's just finished with the ribosome or just going to a ribosome. So these enzymes are going to start nom, 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 eating them. So we've got one enzyme that's going to start at the cap, one enzyme that's going to start at the tail, and they're trying to break down this mRNA so you can use it again. And this is part of the reason why we have the cap and the tail, because here's a little Pac-Man eating away at this poly A tail. Well, guess what? If that tail is long enough, this Pac-Man can eat away at this tail, and that's cool because the ribosome is going to be down here. Ribosome is going to be translating, and by the time the ribosome's done, this enzyme isn't done eating the poly A tail, okay? Once this piece of mRNA is done with the ribosome, though, these little Pac-Man dudes are going to totally gobble up this mRNA, and so it's not going to get translated again. Because if the mRNA was there, then it would find another ribosome, and we would make that protein again and again and again and again. But not if enzymes break it down. Okay, so these little Pac-Man guys are going to be working on chewing up our mRNAs. Okay. Another thing is that we can literally have proteins that that kind of like block translation from starting. Okay, so that it can help block that mRNA from like getting to that ribosome and attaching and having the ribosome assemble. And so even if a piece of mRNA is made, sometimes it can get blocked, and so that it never gets to the ribosome in the first place. And so obviously then that protein is not going to get made. The protein can be processed in any kind of way, which your book talks about, or your protein can just be broken down by these special things called proteasomes. Okay, so these are just different ways that your cell can have to control the amount of protein in the cytoplasm. So those are the answers. For things like epigenetics and cell differentiation, there's going to be, more on this with your other two assignments. We will also have in class on Monday to go through if there's any lingering questions that you still have after this, bring them to Monday's class and I will hopefully answer them for you.